Hello, welcome back again. In today's session, we will see the instrumentation of UV visible spectrophotometer. The instrument that is used to measure the electromagnetic radiation absorbed by the substance is known as the spectrophotometer. The spectrophotometer, there are two different types of spectrophotometer. Number one is the single beam UV visible spectrophotometer, and number two is double beam UV visible spectrophotometer. And you know that in ultraviolet region starting from what 200 to 400 nanometer, and visible region is 400 to 800 nanometer. Now in this session we will see the instrumentation part of what UV visible spectroscopy. See, this is the block diagram of single beam instrumentation of UV visible spectroscopy. It consists of 5 different parts. Number 1 is source, right? This is the source. Number 2 is monochromator. Number 3 is sample tube. Number 4 is detector. And last one is the recorder. This is the block diagram of single beam instrument. In double beam instrument, there is only change. Here is what sample, as well as we can put the reference also in double beam instrument, right? Now we will see the instrumentation part. Number 1 is source. In UV visible spectroscopy, a source of light, that is tungsten filament lamp, is used as a source of light in visible region. In visible region, that is 400 to 800 nanometer region, we can use what? Tungsten filament lamp. And hydrogen lamp as well as deuterium lamp is also used in what? Ultraviolet region, that is from 200 to 400 nanometer region, right? So source of light, the source of light used in UV visible spectroscopy is for ultraviolet region, that is 200 to 400 nanometer. We can use what? Your hydrogen lamp or deuterium lamp is used, and in 400 to 800 nanometer region we can use what? Tungsten filament lamp is used as a source of light. So this is the first part. Now the second part is what? Monochromator. What is monochromator? Monochromator is a device. It can convert polychromatic radiation into what? Monochromatic radiation is known as what? Monochromator. Monochromator consists of what? Suppose this is the source of light, right? Then this source of light, again monochromator. Here is entrance slit, exit slit, right? Here is what? Lens or mirror, and here is what? Dispersing medium or prism, right? So monochromator, it consists of three different parts: entrance and exit slit, lens or mirror, or dispersing medium or prism. Now what is the role of entrance and exit slit? The role of entrance slit is to restrict the unwanted radiation. This is the role of what? Entrance slit. The role of lens or mirror is, that is to focus the beam parallel onto the sample. This is the role of your lens or mirror. And the dispersing medium or prism is to isolate the polychromatic radiation. This is the role of what? Your dispersing medium or prism, right? So I will again repeat: monochromator. Monochromator is a device which can convert polychromatic radiation into monochromatic radiation, is called as what? Monochromator. Monochromator consists of three different parts: entrance and exit slit, lens or mirror, and dispersing medium, right? Then third one is sample tube, or we can say sample cuvette or sample holder. Actually it is made up from what? Glass or a special type of glass, that is what quartz, it can be used. So its internal diameter is around 10 centimeter is the internal diameter of what your sample tube. The role of sample tube is what, to put the sample as well as to put the reference solution into the spectrophotometer. This is the role of what, your sample holder or sample tube. Then fourth one is what, detector. What is the detector? Detector is a device which can convert radiant energy into the electrical energy, is known as what, detector. In UV visible spectroscopy, different types of detector is used. Which one? The photovoltaic cell, phototube, photomultiplier tube. This type of detector is used in UV visible spectroscopy. The detector is a device. It can convert radiant energy into the electrical energy, is known as what, detector. Different detector is used in UV visible spectroscopy, that is photovoltaic cell, phototube, photomultiplier tube. These are the used as a detector in UV visible spectroscopy. And last one is what, your recorder. So recorder, actually it is the display device, right? So absorbance, right, and wavelength, it can be plotted on what, your xy plot. This is the role of what, your recorder. What is the role of the recorder? Recorder, actually it is the display device. Absorbance and wavelength, it can be plotted on what, your xy plot. This is the role of what, your recorder, right? So I will again summarize. Instrumentation of UV visible spectroscopy, it consists of the 5 different parts: source, monochromator, sample holder, detector and recorder. This is the block diagram of what, single beam instrument. And there is a only change in the double beam instrument is what, along with the sample we can put the sample solution also. Okay, now the next important point is application of UV visible spectroscopy. What are the applications of the UV visible spectroscopy? UV visible spectroscopy, it is useful for the qualitative analysis. It is useful for the quantitative analysis. Qualitative analysis means what? Which constituent present into that particular sample. Quantitative analysis means how much quantity of that constituent present into that particular sample. That's why this UV visible spectroscopy, it is useful for qualitative analysis, quantitative analysis. It is also useful for the detection of the impurities present in the sample, detection of the chromophoric group present in the sample. Then it is useful for identification of cis and trans isomers. Cis means what? On same side, and trans means what? At opposite side, right? So therefore it is also useful for identification of cis and trans isomer. Again, it is useful to study the kinetics of the chemical reaction. It is also useful to determine the molecular weight of the compound, right, by using the Beer's law. It is also useful to distinguish between the conjugated compound, conjugated olefins. These are the applications of what, your UV visible spectroscopy. I will again summarize the application of UV visible spectroscopy. UV visible spectroscopy, it is useful for the qualitative analysis, quantitative analysis. Again, it is useful for the identification of the chromophoric group present into the compound. Again, it is useful to determine the impurities in compound, identification of cis and trans isomers, then to study the kinetics of the chemical reaction, to determine the molecular weight of the compound, right? So these are the different applications of UV visible spectroscopy, right? So today we have seen the two different concepts. Number one is the instrumentation of the UV visible spectroscopy, and the second one is application of the UV visible spectroscopy. So if you have any doubt, you can post your queries onto the discussion forum, right? So we will conclude here. Thank you very much.